And so our first speaker is Simone Castellarin from the University of British Columbia. The University of British Columbia is a new partner at IVES, and we are very happy to have this partner from Canada. So Simone Castellarin is Associate Professor at the University of British Columbia. He's Italian from origin, so he did his PhD at the University of Udine, and then he did he did his postdoc at the UC Davis, and then he was appointed at the University of British Columbia. He teaches grapevine and berry crop biology and food biotechnology. In his lab, he investigates the ripening process in wine grapes and the biological mechanisms that determine fruit composition and wine quality, with a particular emphasis on secondary metabolites. And his research includes hormonal signals and molecular signals. So a lot of his work is about environmental constraints like water deficit and the influence on secondary metabolites. So his talk tonight is the effects of water deficit on secondary metabolites in grapes and wines. And so Simone, the floor is yours.
Hello to everybody. I would like to thank the IVES for this invitation. It's really a pleasure for me to present the results of our studies on the effect of water deficit on secondary metabolites in grapes and wine. So there are hundreds of metabolites that are produced in the grapes, and several of them have a strong impact on the quality of the wines. Secondary metabolites are a group of compounds that are synthesized by the plant for the adaptation to the environment. Therefore, when we have changes in the environment, like for instance, lack of water availability, the plant might respond by modulating the production of some of them. Among secondary metabolites, for instance, we have Phenolics, Anthocyanins that have strong impact on the quality of red wines, as well as we have aroma compounds that affect the quality of both red and white grapes. Today, I would like to present some of our studies that aim to understand how water deficit affect the composition of grapes and wines in a red grape variety, Merlot, as well as a wine grape variety, Friulano, also known as Sauvignon Vert. I would like to say that these studies have been performed by Dr. Stefania Savoy at the University of Udine in collaboration with Dr. Fulvio Mattivi at the Edmund Bach Foundation. These studies we performed where they started in the vineyard, where we applied different treatments, including well-irrigated plants and not irrigated plants. But then we had the winemaking part, where we did small-scale fermentation from the grapes obtained from our vineyard trial, as well as Stefania performed in collaboration with the Edmund Bach Foundation and the Ben Gurion University of the Negev, a very detailed compositional analysis of the grapes and on the wine in order to assess the effect of irrigation treatments and water deficit on the composition of grapes and wine. We also performed some molecular analysis in collaboration with the Institute of Applied Genomics in Udine, but I'm not going to focus on this type of analysis in my talk today. So the first study focused on Merlot grapes and wines. We applied water deficit during ripening time, but actually some deficit was experienced by the plant even before veraison, and was a severe deficit that it can be identified in this graph by the strong decrease in the water potential of the plants. And obviously we compared these plants and fruits exposed to severe water deficit with plants and fruits of that were exposed to that were well irrigated, so they were exposed to no deficit. Here is an example of one of our major results. In this complicated graph, we see a long list of metabolites that include mostly phenolics compounds. And in these boxes, we can see if the specific metabolites were increased in red color or decreased in blue color under water deficit. And on the top, you can see 2011, 2012 are the 2 seasons where we performed the experiment. And those numbers, 30, 44, 60, etc., identify the day after antithesis where we collected the samples. So if we focus on 115 here and 106, those days are the harvest time in 2011, 2012. I'm going to just summarize the results of by saying that we saw a strong and consistent increase of several phenolic compounds, mostly phenolic acids and anthocyanins, due to water deficit in the grapes. But we also saw that some other phenolics, for instance, like flavantriols, that are the monomers that they make the tannins, and stilbenoids like resveratrol, were decreasing concentration. We did also perform analysis on the aroma compounds. I'm not presenting here, but we also see that several aromas were increased in concentration under water deficit. Our molecular analysis that I'm not presenting today indicated that there was a direct role of the water deficit on the biosynthesis of some of these compounds. And so the effects we see in concentration were not only due to reduction in the fruit size that normally increase the concentration in the fruit of those metabolites that are produced in the skin, like the phenolics and many aromas, but were also due to direct effects of water deficit on the biosynthesis of these compounds. When we analyzed the wine concentration, we found very consistent results in terms of increases in anthocyanins, so in the pigments, and the concentration of the pigments in the wine. No effects on the tannins, but also increasing concentration of several aroma compounds. And this was actually consistent with what we found when we performed sensory analysis of those wines. And we saw that there was a very strong effect of water deficit on the color, the intensity, but also the hue of the wines, with more blue nose rather than red nose in wines that were exposed to water deficit, as well as a stronger aroma intensity and particularly more fruity aromas in water deficit, in wines produced from grapes exposed to water deficit than in wines produced from grapes that were well irrigated. So these results were consistent with what also other studies have shown, and that's that when we expose red grape varieties to water deficit, we strongly affect the composition of the grapes and the wine, and we see remarkable differences in the quality of the wine we produce from those grapes. Less was known about the effect of water deficit on white grape varieties. This is an aspect of that has rarely been investigated. And in these studies that Stefania performed, we imposed water deficit to Friulano grapes in 2 seasons, 2011, 2012. And here you can see how the plants that were exposed to water deficit looked like at harvest. So that this was a severe water deficit with the reduction of the canopy growth and reduction of actually the leaf area due to leaf abscission. This was observed at harvest time. And those vines were compared to well-irrigated vines. And you can see on the top how the well-irrigated vines had a healthy canopy. Well, we did, similar than before, a large profiling of dozens of secondary metabolites, phenolics, carotenoids, aromas. All these compounds have strong effects on the quality of grapes and wine in general. But I'm going to focus on the major results we found from this study. And one thing, we confirmed that by applying water deficit also to a white grape variety, we can increase phenolics. And these phenolics are phenolic acids, Obviously, we have no anthocyanins, no pigment in the white grape, but other phenolics are produced and were increasing concentration by water deficit. But also we observed something interesting, particularly at harvest time in 2012, at 93 days after anthesis. This group of compounds here on the bottom are aroma compounds, and particularly several of them belong to the class of terpenes, that is a class of metabolites that affect the quality of wines like Riesling, Muscat, Gewürztraminer, and have floral notes and give floral notes to those wines. So we thought it was interesting. We know that Friulano does not accumulate high concentration of these compounds. And in fact, from this graph, you can observe how in controls, that is this full dot here, we have very low levels of alpha-terpineol. That is an example of one of these terpenes that are produced in the grapes. So in control, we have a flat line here. But when we expose water deficit during ripening, we observe a strong increase in the concentration of alpha-terpineol, and we had similar results, for instance, for nerol and for linalool, other terpenes. So we learned that we can stimulate the biosynthesis of these compounds by applying water deficit to a white grape. So we found also an interesting result, that was that in our wines, we had higher concentrations of these compounds. And so we thought that by having higher concentration of these compounds in in wines produced from deficit irrigated vines, we could increase the aroma of the wines. However, despite we found consistency between the concentration of these compounds in the grapes and the wines, so an increased concentration due to water deficit, when we analyzed the sensory features of the wines, we found limited differences between wines produced from well-irrigated irrigated vines and wines produced from deficit irrigated or water deficit vines. So what we found was just a darker or a more intense pigmentation, so yellow pigmentation and more intense body of those wines, but we found no significant effects on the aroma. Now, Friulano produces low levels of these terpenes aromas. So we thought that what we learn from a fundamental side, that is that water deficit can stimulate these aromas, maybe have some implication on the applied side if we apply this water deficit to varieties that are accumulating large amounts of terpenes. And so by applying water deficit, we can further increase the concentration of these terpenes and maybe improve the quality of these wines. So this is how we came with the last study I'm presenting today, that was performed by Eugene Kovalenko here with me at the University of British Columbia in Canada. The study was performed in the Okanagan Valley, one of the major regions for wine and grape production in Canada, and the warmest and driest region that we have here in Canada, where we produce grapes. In this region, we have to sustain grape production via irrigation throughout the season, because the region is characterized by very low precipitation and also by sandy soil. So we have to irrigate weekly or even more intensively during the season to sustain the production of grapes. We focus our study on Gewürztraminer, a variety that is characterized by those aromas, terpenes, that I was mentioning before, that they confirm floral notes to the Gewürztraminer wines. In this study, though, we did not apply only water deficit like I've shown before. We targeted specific deficit levels that were what we call moderate water deficit levels. So deficit levels that are not severely affecting the yield and the growth of the plant, and are actually carefully managing the vineyard by assessing the water potential of the plant and by tuning irrigation accordingly to maintain the plant under stress, but not under severe water stress. So, moreover, we consider different treatments, irrigation treatments. We apply stress before veraison, so we call early water deficit. We apply stress after veraison, we call late water deficit, and we apply this moderate level of stress throughout the whole season. We assess the effect of these moderate water deficit on the canopy growth, canopy physiology, yield, and also fruit composition. I'm going to focus only on two main results of this study. I'm going to present only data on yield and on the terpene composition of this fruit. Here is an example of plants that have been exposed to this moderate water deficit throughout the whole season. Well, yield was affected by these moderate water deficit treatments. It was affected, though, if the treatments were exposed to early water deficit before veraison or prolonged water deficit throughout the season. But interestingly, when we applied water deficit only during ripening time, so from veraison to harvest, we have no significant changes in the yield of the plants compared to the irrigated control. The irrigated control is a standard practice that growers are adopting to grow Gewürztraminer vines in the area. So by saving basically 38% of water, so withholding irrigation or carefully managing irrigation from veraison to harvest, we actually had no effect, no significant effect on the yield of those plants. And this was consistent across the 3 years of the study that we performed. The other result I would like to show is that beside other compositional observation we did on sugars and acids, we focus on the concentration of terpenes because, as I say, those are important aromas for Gewürztraminer. And we found an interesting result that some terpenes, specific ones like geraniol, that is the major terpene accumulating Gewürztraminer, and citronellol were increased in concentration by late water deficit. So that was interesting because that late water deficit was also the treatment that was not affecting yield, and here was increasing the concentrations of important aromas for Gewürztraminer wine. Now, our expectations are that potentially this might increase also the aroma of wines produced from these grapes. However, our studies did not imply in this case the production of wine and sensory analysis of the wine. But we think it's worth exploring further these studies by performing in the future both chemical analysis and sensory analysis of the wines produced from grapes exposed to such treatments like late deficit. So as a conclusion of my talk, I would like to summarize that we know that water deficit affects the production of terpenes and phenolics. So tend to increase the synthesis of several phenolics and several aromas that we know are we call terpenes, that are important because they give this floral and also fruity notes to several varieties. We know that if we apply regulated deficit irrigation, so targeted levels of moderate water deficit, we can improve the concentration of some of these terpenes in grapes, so some of these aromas in grapes. And if we apply this moderate water deficit late in the season, so from veraison to harvest, we have no effects or limited effects on yield, but we can save water. This is important for the production of grapes in areas where irrigation is needed. We know and we have learned that strategies that imply water deficit are suitable for red grape varieties. From these studies, we are learning that they might be suitable to some extent also for the production of some white grape varieties. However, we need to perform more studies to understand what is actually the real effect of these strategies on the wine sensory features. So in conclusion, I would like to thank all the people that were involved in these studies, but particularly Eugene Kovalenko and Stefania Savoy, the two students that performed most of the analysis that I've been showing today. I would also like to thank our industry partners like Andrew Perrer Limited and the BC Wine and Grape Council, as well as several funding institutes that supported these studies. Well, I would also like to thank the International Viticulture and Enology Society that we had the honor to join recently as the wine research center of the University of British Columbia. And I would like to invite you to have a look to our website to learn more about the type of activities we do now in the grape and wine context here at the University of British Columbia. Thank you very much for your attention, and I'm very happy.
Hello. Well, thank you for this very nice talk, Simone. I saw there were a few questions, but I will start with my questions. The fact that water deficit is favorable for red wine quality, in particular because it enhances the synthesis of phenolic compounds, and in particular anthocyanins, is well documented. But I think your research fills an important gap in the literature. There's very little work published on the effect of water deficit on white grapes. And what you show is that water deficit in white grapes can also be favorable to the quality of white wine, which is interesting for our understanding, both in areas in the world where the vines are not irrigated, to understand the vintage effect, that maybe dry vintages can also be favorable for white wine quality. And we know, for instance, that Alsace is a very dry area and it produces very aromatic wines. So that can be, a link can be made with your research. And for countries where the vines are irrigated and areas where the vines are irrigated, this opens interesting perspectives for both saving water and producing high quality wines, in particular with irrigation strategies that not only save water, but don't affect the yield. Now, most of your research focus on the terpene family of aromas. Do you have any idea of what the effect of water deficit on other aroma families like volatile thiols or norisoprenoids? Do you have any results? And if not, do you plan in the future to work on varieties that have other families of aroma compounds, in particular whites?
Yes. Thank you for the question. Can you hear me well?
Yes, sure.
Okay, thank you. We do not have currently results about the impact of these strategies on other classes of compounds, like you mentioned thiols. I could include also methoxypyrazines. We haven't used varieties that are characterized by those aromas. However, we are planning to investigate more on that. Particularly, I think we'll work more on the methoxypyrazine size because, well, in the viticultural areas I've been describing in the Okanagan Valley in British Columbia, in the south of the valley, we grow, well, varieties like Cabernet Sauvignon, Merlot, Cabernet Franc. And so these will be important for the industry. However, that's more on the red grape size. We have interested in Sauvignon Blanc as well. We know the thiols are important there. In our laboratory, unfortunately, at current state, we cannot analyze thiols in grapes and wine, but it is definitely something we see in the future as important to do for us.
There was another question from Eduardo Constantini. We know that there's an interaction between nitrogen and water deficit, and if vines are under water deficit, it may change the nitrogen uptake in the grapes. So there can be an interaction between the effect of nitrogen and water deficit. And we know from the literature that there's also a big effect on nitrogen on aroma compounds. In your studies, did you control the nitrogen status of your vines? And what about the possible interaction between nitrogen and the water status on those aroma compounds?
Yes, so I agree that there could be there is a role of nitrogen, and there are several publications, including some publications that they actually have assessed this interaction of nitrogen and water and irrigation levels and water deficit. In our study, we did not we did control nitrogen because we use only one. We use the standard, I will say the standard fertilization protocols that both the experimental farm at the University of Udine was using, as well as our industry partner here in British Columbia that hosted the field. So in our case, those are the mineral nutrition is kind of a standard protocol, and all our plots was the same. So when I say we control that, is because we were sure that we had the same application. How much it was absorbed, we did not control that in our study. So
it would be worse controlling, but even with applicating the same amount of nitrogen,
it could be
water deficit or without water deficit can maybe possibly absorb nitrogen at different rates. So that's interesting control.
Yeah, this is true. Yeah.
Also, what makes the studies, of course, very complicated is that if you manipulate the water status, then also you have a different microclimate, because as we saw in your pictures, if your vines are under water deficit, then you have...
I can comment on that, yeah. I can comment...
The berry temperature.
Yeah, I can comment on that, and I strongly agree. Actually, we had a focus on that, because the first... you're right, in the first study I've been showing, particularly, you could see that the canopy, the first study I've been showing in Udine, the second study in Udine, so the Friulano study, you could see the picture of a canopy that was well irrigated versus a canopy that was exposed to severe water deficit. And you could see how at harvest, the exposure of the cluster can be and is actually affected. We do investigation of point quadrat, different type of analysis to characterize that. So it was affected. And so that was a concern, because I would like to actually remind the audience that another important factor that affect the production of terpenes is light. So there are studies that are showing that light exposure affects the biosynthesis of these compounds. So in our case, there was a question about, is water deficit affecting the biosynthesis or directly affecting the production of terpenes, or is an indirect effect due to the lighter canopy and more exposure of the cluster? So I think this is a very important thing. But in the second study we did, the study that I showed on Gewürztraminer, we actually did decide to apply leaf removal on all the treatments in order to characterize the canopy, to have a very similar canopy. We also perform point quadrat analysis, leaf area analysis. I didn't show them because of the time, and also temperature checks on the different clusters. I must say that our treatments, the conditions, the microclimate around those clusters were pretty similar in well irrigated and moderate and plants that were exposed to moderate water deficit. So the last study I presented is where we tried to control most of the other factors, talking about light exposure and cluster temperature, and we can say that they were quite consistent among treatments. Sorry, you are on mute, Keith.
Sorry. There was a question from Damien Martin, who asked, are your comparisons of fruit wine composition made at similar sugar maturity? And were there differences in primary metabolites at harvest?
Yeah, so this is a good question. We have been performing for several years this type of studies, and we have varieties that basically respond with different levels of sugars and acids at harvest versus other that they are not affected so much in those components. In our study, for instance, Merlot, we had no significant difference of sugars and titratable acidity between our treatments. In the study, and the wine were produced at harvest, so there were no differences. In the study on Tokai Friulano, we had a slight increase in sugar concentration in the plants that were exposed to water deficit, and this increase was about half a Brix, and we actually had we produced the wine with these two different levels. Just what was kind of the commercial harvest. The titratable acidity I thought was similar, so there was a significant increase, but it was a slight increase, and it was not possible for us to really, it was a matter of few days to distinguish or to follow this. In the study in Gewürztraminer, we didn't produce the wine, but the late water deficit did not affect sugar levels.
Okay. There's a question from Thierry Simoneau, who asked if the increase in terpenes was just due to water loss, or was there also more accumulation if you express it on a per berry level?
Yeah, that's a very, very good question. So in our studies, we tend to express both concentration and per berry. And these increases were also per berry. And in fact, we think it was not due to reduction in berry size. And in fact, the late deficit treatment did not significantly affect the size of the berry. So this was in the study in Gewürztraminer. The study in Tokai Friulano, well, we had only expressed the values in concentration, but we think that there is a direct effect on the biosynthesis, because we have seen that many genes that are directly involved in the biosynthesis of terpenes were strongly upregulated. So this is how actually this thing started. We started from the molecular side, where we saw the pathway being upregulated, and then we look at the concentration of the compound.
I think this is a very important point that you make, and this joins a question also which Mario Pezzotti asked, is because you look also at the genes, the gene expression. If you can find an effect, you can separate a direct effect from an indirect effect. And that is very important in doing such, compared to just doing trials where you only measure fruit composition and wine quality. If you add gene expression, then you can be sure that the possible effect of water deficit is also owner of the genes that control those aroma compounds. So there's time for one or two more questions. So I would like to invite Julien Dumerc on the floor. Maybe you can have a selection from one or two questions from the audience, and then we will have to move to the next speaker.
Thank you very much, Kees. In fact, thank you to the audience for all the questions asked. I will be your representative today, but you already have a good one with Kees. So maybe one question from Lionel, and then we will move to the next speaker. Have you observed a difference in the levels of disease or pests between the different modalities of water deficiency?
Yeah, that's an interesting question. Like we try to control diseases, we have chemical control, and we were lucky that we didn't have major diseases. However, I must say that in the several years we performed these type of studies, particularly on Merlot, the ones we did, I presented 2 years of data, but during my PhD earlier on, we did similar studies. And we saw that when we had in control actually vines, we had higher levels of powdery mildew, higher levels of, actually sometimes we had some powdery mildew infections, but that I think is more related to the canopy and the microclimate of the canopy. We have like the microclimate of the canopy when you don't apply like treatments like I said at the end, where you expose all the clusters, might be significantly different when you apply some moderate or severe water deficit versus protocols that imply, I would say, no water stress. So the canopy grows vigorously and the clusters are more shaded, and there is higher humidity inside, so you can have a higher I think more issues with diseases. Luckily in our studies we could compare our wines and grapes because we didn't observe any remarkable infection. I also want to add just briefly that we with those molecular work that we have done, and then we follow up with another study I'm not presenting, we did observe that several genes that are related to the production of waxes and in the cuticle and of the berries were affected. And we follow up with a study that was published last year, and that shows clearly that water stress changes the composition of the waxes, and also a bit the structure. We have nice microscopy images. I invite you to maybe to have a look if you are interested in this topic. We thought it was more related to transpiration, and we studied the changes in transpiration where we did not observe significant difference, but we think that might have an impact also on, it could be also related to higher or different sensitivity to pathogens, and this is another study we would like to follow up in the future.
Okay, thank you very much, Simone. Well, if we were in the meeting, I would say give a round of applause to Simone for this very nice presentation. In any way, this is exactly the sort of research we want to promote, a very high-level scientific research, but with practical outcomes for professionals.